Usually when you picture advanced chemical manufacturing, you know, the expectation leans really heavily toward this massive industrial scale.
Right, yeah, you envision these towering steel refineries.
Exactly, like miles of pressurized pipes, extreme heat, and engineers operating these million-dollar control panels just to force, stable molecules to react.
Because for the last century, I mean, that has basically been our entire paradigm. We associate chemical synthesis with heavy industry.
We do,
It
yeah.
requires immense energy input, highly volatile solvents, and, well, catalysts crafted from rare earth metals just to break very specific atomic bonds.
But the moment you step into the world of evolutionary biology and forest ecology, that entire brutalist manufacturing landscape just completely vanishes.
Oh, totally. It's a completely different ballgame.
Right. I mean, we are looking at a chemical synthesis engine that operates at ambient temperature in water and with a precision that makes our best industrial refineries look like, like crude sledgehammers.
That's a great way to put it, yeah.
And the crazy part is it's hiding right under our boots.
Literally under our feet.
Yeah. So today we are taking a deep dive into Marasmius rotula, which is commonly known as the collared parachute or the pinwheel mushroom.
Both great names, honestly.
They really are. And our mission today for everyone listening is to uncover exactly how this tiny millimeter-thin organism holds the keys to evolutionary brilliance and futuristic biochemistry.
It's basically the ultimate lesson in biomimicry.
Right.
Because, the most profound scientific breakthroughs, they don't always require synthesizing something entirely new in a lab.
No, sometimes they just require us to look closely at what's already there.
Exactly. Looking
Yeah.
closely at the enzymatic machinery that evolution has spent millions of years perfecting in, well, the most unassuming places.
So we have curated a massive stack of sources for you today to trace this organism's profile.
And it is quite the stack.
Oh, it's huge. We're pulling from historical mycological texts dating way back to the 1800s.
Right, the foundational stuff.
Yeah, and then we are cross-referencing that with the Minnesota Season's ecological database and diving straight into the deep end of 2024 and 2025 biochemical journals.
Specifically, those breakthrough papers from ACS Chemical Biology and Catalysis Science and Technology.
Right, and the synthesis of these sources reveals this just completely fascinating trajectory.
It really does. I mean, we start out with basic field taxonomy, and we end up in the absolute cutting edge of oncology and deep learning AI.
Which is wild.
It is wild. It forces you to realize this isn't just a simple forest decomposer.
No, not at all.
It is a highly optimized biochemical survival engine.
Okay, so before we can examine the specific enzymes this fungus secretes, we really have to look at the physical architecture housing them.
Because the physical structure is a marvel of microscopic engineering all on its own.
It really is. So let's establish the dimensions for you.
Okay, yeah, let's paint the picture.
The cap of the collared parachute maxes out at roughly 2 centimeters in diameter, though the typical specimen you'll actually find on a decaying hardwood log is closer to half a centimeter.
To contextualize that for you, a mature cap easily sits on the surface of a dime.
A dime. That is so small.
Tiny.
And the sources detail the juveniles, the primordia just emerging from the wood as being so small and translucent they appear almost glass-like.
Oh, yeah, they're beautiful.
And apparently you could fit an entire cluster of these juveniles inside an empty acorn cap.
Easily. But what's really striking isn't just the cap, it's the stipe.
The stem, right?
Yeah, the stem. This tiny half-centimeter cap is elevated on a wiry stem that can reach up to 8 centimeters long.
Wow.
Yet that stem is only 1.5 millimeters thick.
That's practically a hair.
Literally. It visually resembles a dark, stiff piece of horsehair, which actually earned it the colloquial name horsehair fungus.
Oh, that makes total sense.
But its morphology isn't static,
Yeah.
It exhibits remarkable phenotypic plasticity.
Meaning it changes based on its environment, right?
Exactly, depending on microenvironmental factors. Like if you observe Marasmius rotula in the spring, emerging on oak or hickory logs, the caps are typically this yellowish-white color.
Okay.
With these deep, navel-like depressions in the center.
Right. The sources mention that. But then it changes.
Yeah, if you observe that same species in the autumn, the structural expression completely morphs.
Wow.
The caps become highly convex, rounding out, and they shift to a light yellow-brown tint.
So it's actively adapting its surface area and pigmentation to the seasonal light and ambient moisture?
Precisely.
That's so smart. But I want to drill down into the mechanics of that stipe really quickly.
Sure, let's do it.
Because you have an 8-centimeter long, 1.5-millimeter thick stem supporting a cap, and the anatomical cross-sections from the literature point out that this stem is entirely hollow.
Right, hollow.
And if you look at the physics of a hollow cylinder versus a solid core, well, a hollow structure provides a really high strength-to-weight ratio.
Which makes perfect sense for saving biomass, right?
Right. But how does a hair-thin, hollow biological cylinder not just buckle under wind shear? Or, immediately succumb to the aggressive damp rot of the forest floor?
That is the million-dollar question.
Yeah.
And it achieves that structural integrity through intense localized melanization.
Melanization.
Yeah. If you examine the stem, particularly moving down toward the base, it transitions from a pale hue near the top to a dark, shiny, almost blackish brown right at the substrate level.
Oh, so it gets darker at the bottom.
Exactly. And that pigmentation is structural melanin.
Okay, wait. We normally think of melanin in terms of, photoprotection against UV radiation, right?
Sure, yeah, in human skin, for example.
But here you're saying it's functioning structurally. It's heavily cross-linking with the chitin and glucans in the fungal cell wall.
Precisely. Melanin in fungi is a highly polymerized, really complex macromolecule.
Okay.
And when it cross-links with the chitin microfibrils in the cell wall, it acts similarly to, well, a carbon fiber matrix.
Oh, wow, like a literal carbon fiber weave.
Exactly. It drastically increases the tensile strength of the hyphae. This allows that hollow stem to flex and, whip around in air currents without buckling or snapping.
That is incredible. But the mechanical strength is only half the battle, right?
Right.
Because the damp leaf litter of a temperate hardwood forest has to be one of the most hostile biological environments on Earth.
Oh, it's absolutely vicious.
It's this hyper-competitive zone of hydrolytic bacteria and antagonistic fungi and, mycophagous arthropods just looking to consume any available biomass.
Which is the second and arguably more critical function of that melanized matrix.
Okay, let's unpack this.
Melanin is highly resistant to enzymatic degradation. By heavily melanizing the base of the stipe, the fungus essentially creates a biochemical quarantine zone.
A quarantine zone. I love that.
Yeah, so the chitinases and glucanases that are secreted by competing soil bacteria, they cannot effectively cleave the melanized tissue.
So they can't digest it.
Right. Furthermore, the polymerized surface creates a hard, unpalatable texture that deters microscopic insects like colembula from grazing on the stem to reach the cap.
Okay, so the fungus invests absolute minimal metabolic energy into a fleshy stipe.
Right, unlike, say, an agaricus or boleta species.
Yeah, the big grocery store mushroom. So instead of being fleshy, it synthesizes this ultra-dense, rot-proofed armored tether.
Armored tether is the perfect way to describe it.
But the true architectural masterpiece is at the top of that tether. The feature that actually gives this species its name, the collarium.
Ah, yes, the collarium.
Because in a standard Basidiomycete, if you look underneath the cap, the spore-bearing gills either run down the stem or attach directly to the top of it.
Right, they intersect.
But Marasmius rotula abandons that entirely.
It creates a physical separation. The gills do not interface with the stipe at all.
Wait, really? They just don't touch it?
Nope. Instead, they attach to a tiny, free-floating ring of tissue, the collarium, that encircles the apex of the stem.
The 20th century mycological illustrator Louis C.C. Krieger actually examined this under magnification, and he wrote, a Swiss watchmaker could not excel such workmanship.
And he was absolutely right.
Because viewed from below, it's a perfectly spaced wheel, right?
Yeah, with the collar acting as the central hub and the gills radiating outward like spokes.
Completely independent of the stem passing through the center.
Right. Mechanically, it functions exactly like a gravity-stabilizing gimbal.
Like a camera gimbal.
The gimbal analogy is the perfect mechanical equivalent. To understand the evolutionary pressure driving this architecture, you have to look at the physics of spore dispersal.
Okay, take us through it.
So, basidiospores are microscopic, and Basidiomycetes rely on a very precise mechanism involving surface tension.
This is often referred to as Buller's drop, right?
Exactly, Buller's drop. It's basically a fluid catapult that launches the spore off the basidium.
Okay.
But once discharged, the spore relies entirely on gravity to drop cleanly through the microscopic airspace between the gills.
Just free-falling.
Right, so it can be caught by subcanopy air currents below the cap.
But if the cap is tilted off its vertical axis even slightly, the spore drops, hits the side of the adjacent gill, adheres to the tissue, and never escapes.
Exactly.
And the entire reproductive effort is completely wasted.
Which is a huge metabolic loss. Now, Factor in the 8-centimeter hollow wiry stem we just discussed.
Oh, I see where this is going.
Because it is so thin and flexible, it inevitably contorts, bends, and twists as it grows up through the chaotic matrix of dead leaves and forest debris.
Right, it's navigating a maze.
Yeah, and if the gills were rigidly attached to that stem, the torsion of the stipe would transfer directly to the cap, tilting the gills out of vertical alignment.
But the collarium completely decouples the cap's orientation from the stem's torsion.
Exactly. The stipe can weave through obstacles, it can bend, but the free-floating collar ensures the cap rests evenly.
So it maintains perfect verticality for the gills.
Yes.
That is insane. It's literally an autonomous, self-leveling suspension system woven entirely out of fungal hyphae.
It is stunning bioengineering.
But, all of this intricate engineering would be biologically useless if the cap just decayed after a few days of sporulation like a typical mushroom.
Right. Which brings us to a metabolic capability that fundamentally alters its lifespan.
It's superpower.
Yeah, the ability to arrest its own biological clock.
Okay, let's talk about the phenomenon of Marcescence.
This was heavily documented by Elias Magnus Fries back in 1838.
Wow, 1838.
Yeah. He was studying gilled mushrooms, and he realized this specific clade completely defined the standard fungal decay curve.
Because most fleshy mushrooms are putrescent, right?
Exactly, putrescent. When their active sporulation phase ends, or if environmental conditions become unfavorable, autolysis begins.
Meaning they self-destruct.
Basically, the internal enzymes degrade the tissue, bacteria colonize it, and it collapses into an amorphous, rotting mass.
A puddle of mush on the forest floor.
Right. But Fries observed that the collared parachute bypasses putrescence completely.
Really? It just doesn't rot.
It doesn't. When ambient humidity drops and the environment dries out, it doesn't rot. It rapidly desiccates, shrivels into this paper-thin, dormant state, and appears totally non-viable.
It looks dead.
It looks completely dead. Yet, upon re-exposure to moisture, it fully rehydrates, expands back into its precise geometric shape, and just resumes its metabolic functions.
That is wild.
Fries actually coined the genus Marasmius based on the Greek word marasmos, meaning withering, precisely because of this capability.
Okay, let me push back on this for a second just to clarify the cellular mechanics for you guys listening.
Sure.
Because if you take a leaf from that same oak tree and let it dry out, it desiccates, right?
Right.
And if you soak it in water, it becomes pliable again. What makes Marcescence different from basic passive desiccation? I mean, rehydrating a dead leaf doesn't make it alive again.
That is the critical distinction. Passive desiccation is highly lethal to standard tissues because of osmotic shock.
Osmotic shock.
Yeah. As water leaves a normal cell, the plasma membrane literally tears away from the cell wall. Organelles collapse, and intracellular proteins denature.
So it's catastrophic structural failure.
Exactly. Re-wetting it simply floods a ruptured, dead cellular husk.
Oh, I see.
Marcescence in Marasmius, however, is an active, highly regulated physiological shutdown.
So it's actually synthesizing osmoprotectants.
Exactly. When the mycelium detects a drop in water tension, the cells rapidly accumulate solutes,
Like what kind of solutes?
likely trehalose or specific polyols. These osmoprotectants stabilize the lipid bilayers and prevent the proteins from unfolding as the water exits the cell.
Oh, wow. So they essentially replace the water molecules.
Right, maintaining the structural integrity of the cellular machinery in this glassy, vitrified state.
Suspended animation.
Exactly. The delicate spore-producing basidia and the complex collarium are preserved in suspended animation.
And then when it rains.
When ambient moisture rises, water re-enters the cells, the osmoprotectants are metabolized or shifted away, and the metabolic engine resumes without catastrophic membrane failure.
That is incredible, and that dramatically shifts the reproductive strategy, doesn't it?
Absolutely does.
Because most standard basidiomycetes operate on a relatively fixed circadian rhythm.
Right, a daily clock.
Once the fruiting body forms, it ejects spores continuously or at specific times of day, essentially gambling that the external humidity will be high enough for that Buller's drop catapult mechanism to work.
And gambling that the spores will germinate once they land. It is a massive energetic gamble.
But Marasmius rotula removes the gamble entirely by ignoring the circadian clock altogether.
Completely ignores it. Its sporulation is strictly moisture-gated.
Like a smart sprinkler system.
Exactly. If the relative humidity isn't optimal for spore germination, it just completely halts spore release, enters Marcescence, and waits.
And because of this regulated on-off switching, the literature indicates a single millimeter-thick fruiting body can survive and intermittently release spores for up to 3 weeks.
3 weeks?
In the ecology of microfungi, where the lifespan of a delicate fruiting body is usually measured in like hours or maybe a couple of days, 3 weeks is an absolute eternity.
It truly is. It maximizes the reproductive return on the initial biomass investment.
Right.
And once you recognize how efficiently this organism manages energy and moisture on the forest floor, you can really start to appreciate how its evolutionary relatives scaled these traits to conquer entirely different microecosystems.
Oh, right. Because some Marasmius species leverage this exact melanized, desiccation-resistant biology to become predators of the forest canopy.
Yes, the spiderweb fungal guild.
Here is where it gets really interesting for you guys. The ecology detailed in the sources regarding tropical and moist temperate forests is just, it's fascinating.
It feels like science fiction.
It really does, because instead of just forming localized mycelial mats in the soil, certain Marasmius species synthesize these highly organized, macroscopic, root-like structures called rhizomorphs.
Right.
And these are thick, heavily melanized cables of hyphae that exhibit apical growth, and, well, they don't stay on the ground.
No, they ascend, they climb up the trunks of trees, anchor themselves, and weave these intricate suspended networks from branch to branch.
Visually, they form massive, black, wiry nets spanning the understory.
Just hanging there. And the ecological strategy here is spatial niche partitioning.
Okay, explain that for us.
Well, the soil level of a forest is an intensely crowded metabolic battlefield.
Yeah.
Every single heterotrophic organism down there is competing for the exact same fallen lignocellulosic biomass.
The dead leaves.
Exactly. By deploying these aerial nets, the fungus bypasses the soil competition entirely.
So it's catching its food in midair.
Yes. It intercepts senescent leaves, twigs, and organic debris midair as they fall from the canopy.
That is genius.
The fungus colonizes and digests this high-quality, undegraded leaf litter while it is literally suspended 30 feet above the ground.
Monopolizing the nutrient stream before the ground-based saprotrophs ever even get a chance to interact with it.
It's a remarkably elegant solution to competitive exclusion.
Yeah.
But it gets better.
Right, because the architecture of these rhizomorphs is so robust that it has actually driven cross-kingdom symbiotic relationships.
GERDs.
Yes. The ecological surveys note that 98 distinct avian species across 27 families, along with small arboreal mammals like flying squirrels, specifically select these Marasmius rhizomorphs as primary construction materials for their nests.
Which makes sense mechanically. The tensile strength of the melanin-reinforced cables makes for a highly durable nest structure.
It's the equivalent of weaving a shelter out of living Kevlar.
Exactly. But the symbiosis goes significantly deeper than just structural utility. The rhizomorph networks are biologically active.
Okay, wait. Active how?
Well, fungi exist in environments heavily populated by pathogenic bacteria, so they naturally secrete a wide array of secondary metabolites.
Including powerful antimicrobial compounds, right, to defend their territory.
Right. So when a bird weaves a nest out of active Marasmius rhizomorphs, it is inadvertently utilizing premedicated antibacterial nesting material.
Mind blown.
Yeah. The nest effectively acts as a prophylactic shield for the nestlings, suppressing the proliferation of detrimental bacteria or ectoparasites that commonly plague avian nurseries.
And in exchange, what does a fungus get?
The nest acts as a nutrient sink. The nitrogen-rich avian excrement, along with the continuous influx of canopy debris brought in by the birds, provides a highly concentrated, localized food source for the fungal network.
So it is a dynamic, mutualistic ecosystem suspended in the trees.
Yep.
And to compound the complexity of this genus, Marasmius rotula also exhibits bioluminescence.
Ah, foxfire.
Foxfire. It is one of the fungal species responsible for this phenomenon, where the actively growing mycelial networks emit a continuous, eerie green light from within the decaying wood matrix.
Yeah, the specific evolutionary advantage of fungal bioluminescence is actually still actively debated in the literature.
Really? What's the leading theory?
A leading hypothesis suggests the light emission at 530 nanometers is precisely tuned to the visual spectrum of nocturnal fungus gnats or specific beetles.
Oh, to lure them in.
Exactly, to lure them to the active site to aid in the mechanical dispersal of spores.
That makes sense. But regardless of the ecological signaling, the bioluminescence requires the oxidation of luciferin by a luciferase enzyme, right?
Yes, which is a very energetically expensive process.
And the fungus can only afford that massive energetic output because its primary metabolism is just devastatingly efficient.
Which brings us to its role as a white rot fungus.
Right. Marasmius rotula is a white rot fungus.
Which is a profound metabolic classification because in the biosphere, plant biomass is primarily composed of cellulose and hemicellulose.
Which are easily digested sugars, right?
Right, but they are encased in a complex, highly recalcitrant three-dimensional polymer called lignin.
The hard stuff.
Yeah, lignin is essentially the biological plastic of the plant world. It provides the rigid structural integrity of wood and protects the cellulose from microbial attack.
And the vast majority of organisms on Earth, including most bacteria and a lot of fungi, completely lack the enzymatic machinery to break the carbon-oxygen bonds in lignin.
They can't touch it. If white rot fungi didn't exist, the carbon cycle would literally halt. Dead timber would just accumulate indefinitely.
That'd be buried in logs.
We really would.
Yeah.
Marasmius rotula operates as a selective white rot decomposer.
Meaning what, exactly?
It doesn't just indiscriminately blast the wood with enzymes. It secretes highly specialized extracellular oxidoreductases that selectively depolymerize the lignin barrier.
So it precisely cuts away the plastic wrapper to get to the candy.
Exactly. Just enough to access and hydrolyze the energy-rich cellulose trapped inside, without completely obliterating the structural integrity of the wood matrix right away.
And to execute that highly selective depolymerization of lignin, which contains incredibly stable, unactivated carbon-hydrogen bonds, this tiny glowing parachute uses a specific set of microscopic chemical tools.
Right.
And this brings us directly to the core of the biochemical revolution we mentioned at the start.
This is where it gets crazy.
Because the exact catalytic machinery this fungus uses to digest wood has recently triggered a total paradigm shift in advanced industrial and pharmacological chemistry.
To truly grasp the magnitude of this shift, we have to look at how humans handle complex chemical synthesis and detoxification.
Okay, let's look at it.
Which brings us to the human liver.
The liver.
Yeah, and a superfamily of enzymes known as cytochrome P450 monoxygenases, or CYPs.
Right, CYPs are the absolute heavy lifters of human pharmacology. When you ingest a xenobiotic compound, a pharmaceutical drug, or a toxin, your liver relies on cytochrome P450 enzymes to process it.
Exactly.
And they achieve this through one of the most kinetically challenging reactions in all of chemistry, right?
Yes. The selective insertion of a single oxygen atom into an unactivated carbon-hydrogen bond.
Okay, explain why that is so hard.
The difficulty of that reaction cannot be overstated. A standard CH bond has a very high bond dissociation energy. It is exceptionally stable and chemically inert.
So it doesn't want to react.
It really doesn't. In traditional synthetic chemistry, if you want to break that bond and insert an oxygen atom to functionalize the molecule, you typically have to resort to really aggressive, non-selective methods.
Like what?
You need extreme temperatures, high atmospheric pressures, harsh acidic conditions, or the use of heavy metal catalysts like palladium or osmium.
Which sounds awful.
It is. It generates a massive amount of toxic waste and unwanted byproducts.
Yet the CYPs in our liver perform that exact oxygen insertion with perfect stereochemical precision.
At 98.6 degrees Fahrenheit.
In a watery environment, without causing radical damage to the surrounding cellular tissue.
It's a miracle of biology.
It really is. And because they are so efficient, the pharmaceutical and fine chemical industries have spent decades trying to harness cytochrome P450 for industrial-scale biocatalysis.
We want to use them in bioreactors to manufacture complex drugs.
Right. But there is a massive logistical hurdle.
The cytochrome P450 problem.
Yes. Let's talk about the problem.
The problem is that CYPs are biologically delicate and incredibly high maintenance.
High maintenance how?
First, they are integral membrane proteins, heavily anchored into the endoplasmic reticulum.
Okay.
If you extract them from the lipid membrane to use in a reactor, their structural conformation degrades and they completely lose activity.
Oh, so they fall apart outside the body.
Exactly.
Right.
Second, their catalytic cycle is incredibly convoluted. They are not self-sufficient enzymes.
Right. They contain a heme thiolate active site that needs strict regulation.
Yes. To activate the oxygen molecule, the CYP requires a precise sequential delivery of 2 single electrons.
And those electrons don't just appear out of thin air.
No, they don't. The CYP relies on a separate, complex partner protein, a cytochrome P450 reductase, to shuttle the electrons.
Okay.
Furthermore, that reductase needs an electron source, which in biological systems is NADPH.
NADPH, and that is expensive, right?
Exorbitantly expensive. NADPH is a highly complex, unstable chemical cofactor. Using stoichiometric amounts of NADPH in large-scale industrial manufacturing is economically impossible.
To put it in perspective for you, using cytochrome P450 is like relying on a Formula One hypercar for a cross-country delivery route.
That's a really good analogy.
Like, yes, it is the most precise, highly engineered machine available, but it requires a pristine environment, a dedicated pit crew of helper proteins to just keep it running, and astronomically expensive custom fuel, the NADPH, to even start the engine.
Right. It is brilliant in vivo, inside the body, but completely unscalable in vitro in the lab.
So the industry desperately needed a catalyst that performed the exact same highly selective oxygenation chemistry as the CYPs, but was built with the rugged simplicity of a tractor engine.
And for a long time, the consensus was that such an enzyme might not even exist, or at least would be incredibly difficult to engineer from scratch.
That was until researchers isolated the secretum of Marasmius rotula and identified MroUPO.
Ah, yes. Marasmius rotula aromatic peroxygenase.
This specific enzyme is the keystone of our entire deep dive today.
It really is the hero of the story.
Because the fungus secretes MroUPO into the extracellular environment to selectively oxidize the lignin we discussed earlier.
Right.
But structurally, it is a revolutionary hybrid. It bridges the gap between classic rugged peroxidases and the delicate, precise cytochrome P450s.
And the key to MroUPO's incredible value is the concept of the peroxide shunt.
Okay, break down the peroxide shunt.
Instead of relying on a reductase partner protein and an expensive electron donor like NADPH to activate molecular oxygen.
Like the human liver does.
Exactly. MroUPO directly utilizes hydrogen peroxide, H2O2, as both the oxygen source and the electron acceptor.
So it completely bypasses that entire complex electron transfer chain.
Totally bypasses it. You have this self-sufficient, single-component enzyme.
Wow.
It possesses a highly constrained active site pocket that mimics the stereospecificity of a CYP, but to fuel the reaction, all you have to do is slowly drip in cheap, commercially available hydrogen peroxide.
That is insane. So the enzyme uses the H2O2 to form compound I, which is that highly reactive iron oxo species at the heme center, right?
Yes.
And then that abstracts a hydrogen atom from the target substrate, leading to the highly precise insertion of the oxygen atom.
Exactly. It's so clean and simple.
Wait, I have to ask a theoretical question here, though.
Sure.
Because if utilizing hydrogen peroxide via the peroxide shunt is vastly more efficient and doesn't require the massive energetic investment of NADPH and reductase proteins, why did mammalian livers evolve to use the incredibly convoluted cytochrome P450 system? Wouldn't evolutionary pressure favor the simpler, cheaper pathway for us too?
That is a phenomenal question.
Yeah.
It really comes down to spatial regulation and oxidative stress.
Okay, how so?
Hydrogen peroxide is a highly reactive oxidant. If human liver cells relied on high intracellular concentrations of H2O2 to drive their detoxification pathways, the ambient leakage of hydroxyl radicals would cause catastrophic oxidative damage to our DNA and cellular proteins.
Oh, wow. So we'd basically poison ourselves.
Exactly. The human liver uses the convoluted NADPH-dependent CYP system precisely because it allows for absolute tight regulation of the oxidative process within the delicate intracellular environment.
Ah, so the CYP system trades energy efficiency for safety.
Right. But the fungus doesn't have that problem.
Because it's an extracellular enzyme.
Exactly. MroUPO is an extracellular enzyme. The fungus pumps the enzyme out of its hyphae into the deadwood matrix, away from its own delicate DNA.
So out there in the dead leaf litter, the fungus can freely generate hydrogen peroxide to drive the peroxygenase reaction without risking autotoxicity.
Yes. Evolution designed MroUPO to operate in harsh, unregulated environments, which makes it the absolute perfect candidate for harsh industrial bioreactors.
And the production metrics back that up, right?
Oh, the yields are staggering.
Because MroUPO evolved to be actively secreted, scientists can culture the fungus in liquid media and harvest the enzyme at unprecedented rates.
Yes, the sources highlight that MroUPO can be produced at yields exceeding 445 milligrams per liter.
445 milligrams per liter. That is huge.
It's roughly 40 times higher than the expression levels of previously studied peroxygenases, It is a rugged, highly scalable, self-sufficient chemical factory.
So what happens when we extract this highly evolved lignin-degrading enzyme from the forest floor and apply it to modern pharmaceutical development?
Well, the 2020 and 2024 biochemical studies focus precisely on this transition. They utilized MroUPO to synthesize human drugs, most notably cyclophosphamide, or CPA.
Right, CPA. CPA is a foundational alkylating agent used extensively in chemotherapy regimens and as an immunosuppressant for severe autoimmune disorders.
It is a very common drug.
But CPA is classified as a prodrug, meaning when a patient receives an intravenous infusion of CPA, the molecule itself is pharmacologically inert. It doesn't interact with the cancer cells at all.
Right, it relies entirely on hepatic bioactivation.
Hepatic, so liver.
Yeah. When the inactive CPA circulates through the human liver, specific cytochrome P450 enzymes, predominantly CYP2B6, hydroxylate the molecule. They insert an oxygen atom to convert CPA into 4-hydroxycyclophosphamide.
4-OH-CPA.
Exactly.
And 4-OH-CTA is the critical primary metabolite. It exists in equilibrium with its tautomer, aldophosphamide, which then circulates to the target tissues and undergoes spontaneous elimination to form the actual cytotoxic agent.
Phosphoramide mustard.
Phosphoramide mustard, along with the toxic byproduct, acrolein.
Right, and the phosphoramide mustard then forcefully alkylates the guanine bases in the cancer cell's DNA, creating cross-links that physically prevent DNA replication.
Inducing apoptosis, cell death.
Exactly. The liver acts as the necessary ignition switch.
But this creates a severe bottleneck in pharmacological research.
A massive bottleneck.
Because if toxicologists are trying to evaluate the efficacy of new CPA derivatives, or if they want to test the direct cytotoxicity of the active metabolite on specific human cell lines, they cannot use the stable inactive CPA.
No, they desperately need a supply of pure, pre-activated 4-OH-CPA.
And achieving that through traditional chemical synthesis is an absolute nightmare.
It really is. Synthesizing 4-OH-CPA typically involves fentanyl oxidation chemistry.
Okay, fentanyl oxidation.
Yeah, you mix iron catalysts with hydrogen peroxide to generate hydroxyl radicals. But hydroxyl radicals are wildly unstable and highly unselective.
So they just attack everything.
They attack the CPA molecule randomly. The data shows that standard fentanyl oxidation yields a dismal 3 to 4% of the desired 4-OH-CPA.
3%? That is terrible.
It is, while generating a massive array of useless oxidized side products. It's incredibly inefficient.
Enter the fungal peroxygenase.
MroUPO to the rescue.
Researchers hypothesize that because MroUPO's active site structurally mimics the CYP enzymes, it might be able to perform the exact same bioactivation.
Right, so they introduced the inactive CPA to the fungal MroUPO enzyme in a highly controlled single-phase reaction, slowly feeding it extremely low concentrations of hydrogen peroxide to avoid inactivating the heme center.
And within 60 minutes, the fungal enzyme achieved a 42% conversion yield of the pure activated 4-OH-CPA.
42%. That is a massive leap from the 3% seen in chemical synthesis.
Insane, but more importantly, it operated with extreme regioselectivity, producing less than 3% of unwanted side product.
It performed clean, pharmaceutical-grade hydroxylation.
But, synthesizing the molecule is only half the proof.
Right. The critical question was whether this fungal-derived metabolite behaved identically to the human-derived metabolite in an actual biological system.
And to prove this, the researchers executed a series of really sophisticated cellular assays.
They took the fungal-produced 4-OH-CPA and applied it directly to in vitro cultures of human breast adenocarcinoma cells.
The MCF7 cell line?
Yes, and human hepatoblastoma cells, the HepG2 cell line.
Okay, and what did they find?
They monitored the metabolic response of the cancer cells, specifically looking at ATP depletion.
ATP being the primary energy currency of the cell.
Right. Within a very short exposure time, the fungal-activated drug caused a precipitous drop in intracellular ATP levels.
Indicating rapid systemic cellular failure and mass apoptosis.
Exactly. But they went a step further to prove the precise mechanism of action.
They utilized immunofluorescence to track a specific biological marker known as gamma H2AX.
Yes, gamma H2AXA. When a cell suffers severe DNA double-strand breaks, it rapidly phosphorylates the H2AX histone protein at the site of the damage to flag it for repair.
It's like a flare going up where the DNA is broken.
Perfect analogy. By staining for those gamma H2AXA foci, the researchers could visually quantify the damage to the cancer genome.
And the data definitively showed that the MroUPO-produced metabolite shredded the cancer's DNA with the exact identical efficacy and molecular mechanism as the commercial gold standard drug activated by human liver microsomes.
Identical.
The profound implication here is that an enzyme evolved to degrade decaying oak leaves can perfectly replicate the complex pharmacokinetic metabolism of the human liver in a simple glass beaker.
It is paradigm shifting. This technology fundamentally alters how we screen prodrugs.
It heavily mitigates the reliance on expensive animal models, right?
Oh, massively.
Mhm.
Or the complicated harvesting and stabilization of human liver microsomes just to evaluate a drug's toxicity profile.
And because poor TPO's catalytic pocket is highly versatile, its utility scales way beyond oncology.
Oh, yeah. The enzymatic framework designed to break down recalcitrant lignin is remarkably effective at neutralizing recalcitrant synthetic pollutants as well.
Environmental cleanup.
Yes, the 2024 literature details how MroUPO can effectively degrade 35 of the 40 most dangerous environmental pollutants flagged by the EPA.
Yeah, 35 out of 40. This includes the rapid oxidation of polycyclic aromatic hydrocarbons, or PAH, right?
Yes. PAHs are highly carcinogenic fused ring compounds generated by the incomplete combustion of fossil fuels.
And they are notoriously persistent in soil and groundwater.
But MroUPO's ability to selectively hydroxylate the aromatic rings of PAHs breaks their chemical stability, rendering them susceptible to further degradation by standard soil bacteria.
It also demonstrates exceptional capability in hydroxylating complex agricultural herbicides, such as clomazone and the fungicide mepanoparam.
Right. By using the fungal enzyme to rapidly generate the oxidative breakdown products of these chemicals, environmental scientists can map out exactly how these herbicides will degrade in the watershed over time.
Allowing for rapid toxicity assessments before these chemicals poison an aquifer.
It's a total game changer for environmental science.
We basically possess this universal biochemical skeleton key.
We do.
But the transition from discovering a miracle enzyme to implementing it on a global industrial scale introduces its own set of distinct engineering hurdles.
It's never as simple as just finding it.
Right. The scientific community is currently aggressively iterating on this protein using the most advanced synthetic biology techniques available in 2024 and 2025.
And the primary hurdle they faced with MroUPO was heterologous expression. MroUPO is a homodimeric protein.
Okay, let's clarify that structural constraint for the listeners. A homodimer means the functional enzyme isn't just a single folded polypeptide chain.
Right, it consists of two identical protein subunits that must bind together perfectly to form the active quaternary structure.
Exactly. And in the case of MroUPO, these two subunits are locked together by multiple complex disulfide bridges.
Covalent bonds between sulfur atoms and the cysteine residues.
And these disulfide bonds are exactly what gives the enzyme its extreme stability in harsh extracellular environments, but they make genetic engineering incredibly frustrating.
Oh, a total nightmare.
Because if you take the gene for MroUPO and insert it into a standard bacterial host like Escherichia coli,
the bacteria can transcribe the protein, sure. But the bacterial cytoplasm is a highly reducing environment. It cannot properly form those complex disulfide bridges.
So the protein just misfolds into useless inclusion bodies?
Exactly. So researchers shifted to a eukaryotic host, Saccharomyces cerevisiae, which is common baker's yeast.
Because yeast possesses the secretory pathways necessary for disulfide bond formation.
Right. However, S. cerevisiae tends to heavily overglycosylate secreted proteins.
Meaning it attaches sugars to it?
Yeah, massive sugar chains to the surface of the enzyme, which in MroUPO's case completely shielded the active site and ruined its catalytic efficiency.
The yields of properly folded functional MroUPO were abysmal.
So frustrating.
We had the perfect engine, but our biological factories lacked the precise machinery to assemble it.
But the solution didn't come from forcing the yeast to change.
No, it came from looking sideways on the evolutionary tree, culminating in the 2025 breakthrough involving Marasmius wittsteini.
Right. Researchers hypothesized that if Marasmius rotula possessed this enzymatic capability, closely related species in slightly different ecological niches might possess similar enzymes with slightly different folding kinetics.
So they sequenced the genome of Marasmius wittsteini, a sister species that visually mirrors the collared parachute, but specifically colonizes highly acidic pine needle litter rather than broadleaf hardwood litter.
And when they mined the M. wittsteini genome, they identified a homologous peroxygenase gene, which they designated MroUPO1.
And when they mapped the protein, they found it was 96% identical to MroUPO.
96%. It retained the exact same active site architecture, meaning it possessed the identical capacity to oxidize CPA and degrade PAH pollutants.
But the remaining 4% difference in the genetic code, which equated to just 11 specific amino acid substitutions along the protein's surface, fundamentally altered its expression profile.
Those specific substitutions perfectly optimize the protein's folding kinetics for yeast expression.
MroUPO1 bypassed the glycosylation shielding issues and proved to be perfectly compatible with the state-of-the-art tandem yeast expression system.
Yes.
Let's break down why that tandem system is so powerful, because it solves the two conflicting goals of enzyme engineering: discovery and mass production.
Precisely. The first step, discovery, involves directed evolution. You need to create thousands of mutant variations of the enzyme to find one that works perfectly for a specific industrial task.
And Saccharomyces cerevisiae is fantastic for this because it's genetically highly tractable.
You can easily utilize homologous recombination to generate massive mutant libraries quickly and screen them. But S. cerevisiae is a terrible organism for high-density, industrial-scale fermentation.
So once the researchers used the S. cerevisiae platform to evolve and isolate the optimal genetic variant of MroUPO1, they extracted that specific DNA cassette and integrated it into the genome of a completely different yeast species, Pichia pastoris.
Right, Pichia pastoris. That is the industrial powerhouse.
It thrives in high-density bioreactors and secretes enormous amounts of properly folded protein directly into the culture medium.
By utilizing this tandem system, the 2025 data shows they successfully produced highly active recombinant MroUPO1 at massive titers, reaching 0.7 grams per liter.
0.7 grams per liter. That is an astonishing yield for a complex eukaryotic enzyme.
It really is. By leveraging the genetic diversity of a closely related sister species, they entirely bypassed the homodimer bottleneck.
We have officially entered the era of designer peroxygenases.
We can actively direct the evolution of these fungal enzymes in weeks, scaling production to target highly specific pharmaceutical syntheses or rapidly deployable bioremediation cocktails for specific toxic waste spills.
It's just brilliant. But, the prerequisite to this entire bioindustrial pipeline is discovery.
Always. You cannot extract the genetic data of a novel peroxygenase if you cannot accurately identify and catalog the fungus in the wild.
And this highlights a massive operational bottleneck in modern taxonomy, because distinguishing a species within the Marasmius genus from a morphologically similar but biochemically distinct genus like Mycena relies on minute microscopic differentiations.
Right. Field taxonomy isn't just looking at the color of the cap. It requires an expert mycologist to analyze spore prints, measure basidiospore dimensions, and examine the microscopic morphology of cystidia.
The specialized sterile cells interspersed among the basidio on the gill surface.
Exactly. That level of specialized analysis is incredibly slow and relies on a rapidly dwindling pool of classical taxonomic experts.
And if the cataloging process remains a purely manual endeavor, we face a severe biodiversity crisis. Given the rate of deforestation and microclimate shifts, we are at profound risk of losing vital species before their unique enzymatic profiles are ever sequenced.
To accelerate this, computational biologists have integrated artificial intelligence directly into the taxonomic workflow, specifically utilizing a novel deep learning architecture.
The studies detail the implementation of a CNN-SO model.
Deep learning is frequently thrown around as a buzzword, so let's unpack the actual mechanics of this architecture because it solves a massive flaw in standard AI image recognition.
Okay, let's do it. The first half of the model is a CNN, a convolutional neural network.
CNNs are the foundational architecture for computer vision. When you feed a CNN an image of a mushroom, it applies mathematical filters convolutions to the pixels to extract spatial hierarchies of features.
Okay.
The early layers detect simple edges and textures, while the deeper layers synthesize those edges into complex shapes, like the specific curve of a cap or the attachment point of a gill.
And the CNN can process massive data sets and classify the image with high statistical accuracy.
Yes. But the fatal flaw of a standard CNN is that it operates as a black box.
Right.
The final layer of the network condenses all those complex mathematical extractions into a simple probability score. It outputs, I am 98% confident this image is Marasmius rotula.
But if a mycologist asks the model why it made that classification, what specific biological features drove that mathematical probability, the CNN cannot articulate it.
And in taxonomy, where the burden of proof relies on definitively demonstrating distinct morphological traits, a black box prediction is scientifically useless. You need verifiable interpretation.
Which is why the researchers modified the architecture by replacing the final classification layer of the CNN with a SOM, a self-organizing map.
Right. A SM is an unsupervised neural network technique utilized for dimensionality reduction.
Explain that a bit more.
Instead of just collapsing the data into a single prediction, the SOM takes the highly complex, multi-dimensional feature data extracted by the CNN and projects it onto a two-dimensional visual grid.
Okay.
It organizes the data topologically, meaning images with similar abstracted features are clustered closely together on the map, while dissimilar images are pushed apart.
So by operating as a supervised CNN-SOM hybrid, the AI actually becomes interpretable.
Exactly. When it classifies a specimen as Marasmius, the researcher can examine the specific node on the SON grid where the image was clustered and trace the weights backward to see exactly which visual features the AI prioritized.
The model essentially shows its work, highlighting the minute structural variances in the stipe or the distinct spacing of the collarium that differentiate it from a mycena.
This completely revolutionizes the speed of ecological surveys. It provides field researchers with an interpretable, highly accurate computational tool to rapidly sort and classify massive visual datasets of forest microbiomes.
It dramatically accelerates our ability to map biodiversity, ensuring we can pinpoint and preserve the ecological niches of these hyper-specialized fungi before they are lost forever.
The synthesis of all these fields is simply staggering.
We have traced an incredibly complex narrative arc today.
We really have.
We started by examining a tiny, seemingly insignificant organism on a decaying log, a millimeter thin, glowing mushroom with a heavily melanized armored stipe and a highly engineered free-floating collarium.
We analyzed its specialized physiological ability to enter Marcescence, surviving extreme desiccation and perfectly timing its reproduction to ambient moisture, an adaptation so successful that its relatives utilize it to construct aerial, pre-medicated rhizomorph networks in the canopy.
We then dove straight into its cellular machinery, dissecting how its unique evolutionary mandate to digest lignin resulted in MroUPO,
A hybrid chemical catalyst that perfectly mimics the highly regulated oxidation chemistry of the human liver's cytochrome P450 system, but utilizes simple hydrogen peroxide to operate.
an enzyme that fundamentally democratizes pharmacological testing by efficiently bioactivating prodrugs like cyclophosphamide, shredding cancer DNA in vitro, and simultaneously providing a robust biocatalyst capable of neutralizing highly carcinogenic environmental pollutants.
And finally, we explored how advanced tandem yeast engineering and interpretable AI architectures are actively scaling this microscopic forest biology into global, mass-manufactured industrial solutions.
The overarching through line of this entire discussion reinforces the absolute necessity of rigorous foundational curiosity.
Absolutely. The solutions to our most complex engineering and medical bottlenecks rarely exist in a vacuum. They are often already functioning flawlessly in the wild, optimized by millions of years of selective pressure.
A walk through a damp, decaying forest is quite literally a walk through the world's most advanced, highly efficient chemical manufacturing facility. We merely need the insight to look closely enough at the processes occurring at the microscopic level.
Which leaves you with a final provocative thought to consider as you step outside today.
Yes. We now understand that the metabolic engine contained within a single hollow hair-thin stem of Marasmius rotula possesses the biochemical power to mimic our own liver, bioactivate targeted cancer therapeutics, and degrade the most recalcitrant synthetic toxins on Earth.
Knowing that level of chemical mastery exists in a half-centimeter mushroom, how many other microscopic, hyper-specialized biological engines are quietly operating underneath the dead leaves in your own backyard right now?
What other currently unimaginable chemical solutions are sitting in the soil, waiting for us to develop the curiosity to find them before they fade away entirely?
It fundamentally alters your perception of what is occurring in the soil beneath your feet.
It truly does. Thank you for joining us on this deep dive into the architecture and chemistry of Marasmius rotula. Keep looking closely at the unseen systems around you, keep asking the difficult questions, and stay insanely curious.